So, hello everybody, welcome to today's computational genetics discussion group. Today we have Brian Rice speaking about rapid transfer of knowledge between genetics and breeding for simultaneous trait delivery and genetic gain. He's a Canadian that moved to US to get the bachelor's at Purdue University, and currently Brian is a postdoc researcher at Colorado State University. His training is in plant breeding and quant gen. He has a master and PhD from University of Illinois in quantitative genetics. His current work focuses on bridging the gaps between crop genomic resources and smallholder farming serving plant breeding programs. So Brian, thanks again for being here today with us, and please, whenever you're ready, the floor is yours.
Thanks for the introduction, Ivan, and thanks everybody for being here, regardless of if there was cookies or not. It's been ever since I found the seminar earlier this year. I've been attending it pretty regularly, and it's an honor to be asked to present in it. So today I'm going to talk about my PhD, what is my PhD work, and what is my postdoc work, and will become kind of the basis for my research going forward in my career. And it's a framework that I've developed and is in development, and this one I'm present on today. I'm calling it flywheel genomics. So name subject to change. So just to echo my background, so I do a PhD in quantitative genetics under the guidance of Alexander Lipka, who I believe, if I'm not mistaken, is coming to the Roslin Institute to do a sabbatical. So some of you guys might have the pleasure to meet him. I'm currently a postdoc researcher at Colorado State University, and this is where we reside, right in the foothills of the Rocky Mountains. It's awesome place to it's one of the, maybe like Edinburgh, one of the cool places to do plant genetic resources research, and a cool place to live. So under the guidance of Jeff Morris. And then I've accepted, and I'll be starting as an assistant professor at the University of Nebraska in January. So excited about that. So today I'm going to kind of start us off kind of why I do research, and why I believe the framework that I'm going to present today is important. So the type of breeding and the type of farmers that I'm personally in academia to serve are the smallholder farmers. And so we can characterize a smallholder farmer as typically less than 10 acres or hectares of farmland. Oftentimes, most of the time, they're renting this farmland. They don't own it. And most of the time, they're feeding their family entirely off this land, and trying to make extra money to or feed their own animals, so that they might have milk, or they might be able to sell their animals for extra money. And so their livelihood is very much tied up in their 10 hectares or even oftentimes less of land. In particular, Asia and most of Africa is characterized as smallholder farming, and so just a large proportion of the world especially in these regions, their livelihood relies on being able to farm in this way. So just to narrow in kind of on the Africa region, so this is a very characterized as very arid environment, and because of that, we expect that the effects of climate change are going to actually be even more drastic in these areas as well. These are currently and predicted to continue to be some of the highest population growth on the planet. And this really leaves them, if they're not already, continue to increase their susceptibility to food insecurity. And so this is, again, why I'm in research. Just to narrow in on a crop, the crop I'll be talking about in specific today, but the framework I think I'll present today can work across crops. And so sorghum, one of the most important crops in Africa, as a continent, the estimate I found about 20 million tons per year being produced. It's a plant in the Poaceae family. It's a very beautiful plant. It's got its male and female flowering parts on the head of the plant, and because of this, it's self-pollinating, but it's also actually not too hard to cross-pollinate. It's a great plant to work with. Being in Africa, most of the major stress is drought, water deficiency at different stages of the growing season, but also weeds. The main one is striga, which is also known as witchweed. It chokes out the plant. And then a list of other ones, but basically, just like many crops, we have the yield losses. The difference in these farming systems is that they're very low input, if most of the time negligible inputs. And so in order to actually overcome these abiotic and biotic stresses, the genetic resistance or is needed. Management is less reliable of an option. And so because of this, new adoption of new varieties, sorghum varieties. There are sorghum varieties being developed for Africa constantly, but the adoption is really low. Just as an example, a lot of the farmers still grow land races or that they've been self-propagating for hundreds of years. And then even some varieties that were released in the 90s are still some of the prominent varieties in Africa. The issue being is that a lot of breeding is focused on pushing yields ahead, but the farmers really need these certain traits, or else they don't really accept the new varieties. There are these traits available. The issue being that there's a lack of actually putting the right trait in the farmer preferred variety, there's like the panicle has a ton of diversity too, and so the farmers even prefer just different panicle types, and oftentimes those aren't what's being released to them. So it's just a matter of right trait, right genetic background, and then in the right environment. And like I said, this just low input, and so traits are actually super important here. And a lot of the land races and the older farmer preferred varieties have the traits, but they're not improved. So just to wrap up kind of why I'm in research. I envision where smallholder farmers are, they're able to achieve their own self-sufficiency and food security on this land that they're growing, provide for their family, increase their quality of life. And towards that end, my mission is to help improve breeding for these locally adapted cultivars varieties, and also breed to meet what's going to be rapidly changing environments in these parts of the world, and all parts of the world. So how does this framework kind of work? So if the purpose of breeding, right, is to release varieties, increase genetic gain. It's very simplified, but I think an accurate description of breeding. And then the purpose of genetics at an academic level, it would be, to produce knowledge, right? This is what we do in genetics, and the hope is that this knowledge is helping breeding do what breeding needs to do. And so quantitative genetics, I'm a quantitative genetics, so we'll kind of focus on how genetics does this from a quantitative perspective. And oftentimes to learn about traits, we use mapping populations. And so there's just a diagram showing some recently introduced recombination, maybe like a NAM, a biparental cross or something. And then people often study large diversity panels, and this is just a structure PCA plot showing the structure of sorghum, and so you can see that we have a ton of genetic data for for sorghum, but for other species as well too. Ton of genetic data, ton of phenotypic data, and we can do our association studies. And then also stick in here that genetics also has available to us molecular perturbation, so the molecular side, which is not where I work and is not what I'm going to go into, but it is available to genetics. And just to reiterate, what genetics has available to us from a quantitative genetics perspective, right? We can help with prediction. We can do prediction models. We can learn which prediction models are best and how to predict in new environments. We can do trait genetic dissection, in these, find out how many QTLs, what the QTLs are, how the QTLs behave in different environments. And then, we can also just describe different variance components, that are in these models, that are doing these predictions or characterization. So, to kind of build upon these different populations that are available, this is specific to plant breeding, plant genetics. We have diversity panels, we have NAMs, magics, and then just in more specific biparental crosses, RILs, recombinant inbred lines. So I've sort of kind of characterized these different populations in which and gave them a score of high, low, or medium for these different characteristics. And so mapping power, what's its ability to find a true association. The diversity, this is going to rely on which alleles we can actually discover. The population structure and phenotypic covariates, this is going to kind of determine our level of true and false positive rates. And then finally, the one that I'm most interested in is how, what we learn from these different type of populations, how transferable is that to breeding programs. And so if you discover a QTL in these populations, we know that the stats, what we know about it, most likely going to be population, environment, and genetic background specific, right? And so that means that if we're going to use that information, use that QTL in a breeding program, we have to integrate it if it's not already present in the breeding program, and reevaluate its performance, the stats, revalidate it to make sure it's even a thing that can work. We might even have to evaluate negative pleiotropy, see if that's a thing. Epistatic effects with the new genetic backgrounds it's in. And this inherent struggle to transfer this information to breeding programs comes from because these mapping populations are not breeding populations, right? These mapping populations, I would refer to them as static populations. They are set up once. A lot of work goes into setting them up sometimes. There's diversity panels, people characterize and collect these panels. But they're static. Breeding is a dynamic process. None of these populations are dynamic. If we want to understand the genetics of a trait that matters for breeding, we need to study it in dynamic populations. And so, right, what is a dynamic population, right? For breeding, the dynamic population is this population that's under selection, right? So we have our distribution, our population. The breeder makes a selection, some truncated selection, and shifts the population towards a new optima, or the optima that they're chasing. And then, they can make new crosses, add in new recombination and new alleles into it, right? And so breeding is not this, it's not just engineering new plants. This is an experiment in local adaptation, right? The breeder has a target for local adaptation, and that they're trying to achieve through these methods. And we can use the theory of selection, of adaptation to guide how we might study a trait, how we might map a trait in a breeding population. And so conceptually, how might that look? Well, if we have an allele that's a favorable allele and is under selection over time in a population. When the allele frequency is low, the tool that'd be most useful would be linkage mapping. So you might have to screen and identify it and then create your aside biparental. And so we haven't gotten away from that, but we are using the breeding material as the allele frequency becomes more common. We're able to use GWAS in a population to look for common alleles. And then as the allele heads towards fixation, as the breeder, this is the breeder's goal, with selection scans to identify it are going to be more useful. So we did this, and I'll kind of discuss this, what this kind of looked like. So we worked with a breeding program in Haiti, which is an entire side presentation of how difficult it is to breed right now in Haiti. But oftentimes their sites they're cut off by gangs, and they can't. We'll be discussing data, and they're like, oh, that's at this site, but I can't get there unless I go take my motorcycle and go through the mountains because I can't drive the road because it's cut off by gangs. So that's a whole side conversation, but it's pretty intense. And Gael Pressoir, native of Haiti, trained at University of Cornell, is doing an amazing job out there. Brilliant scientists, and none of what I'm going to show you today would be possible without him. So in Haiti and other parts of the world too, but there was a large biotype shift in the sorghum aphid, and which wasn't really an economic issue before, all of a sudden became an economic issue. And I'll just show you, this is an image of aphid infestation. You can see how intense it is. They're having a great time. And this is a susceptible variety right next to a resistant variety. And there's no survival. In Haiti, for example, in order to grow sorghum that isn't resistant, you have to spray 3 times a year for aphids, if not more. And it's just not feasible. Haitian farmers are also smallholder farmers, and that's just not feasible. It's not even feasible in the breeding program at a large scale. And so once this biotype shift came here at the red line here, right, just production plummeted in Haiti. Gael's program went under a very hard selective sweep, not even by his own choice, just everything that wasn't resistant in the program died. And then he continued to breed with that population, and continued. He's the only breeder in Haiti, so this yield gain, this production gain is fully attributed to his breeding program. And so we know that right here at 2, there was a hard selective sweep. And what can we do? We can do a selection scan to identify what he actually selected on. And actually for this resistant to this aphid, there was previously only one known QTL that was identified in a biparental population by a Chinese group. And sure enough, that's what was under hard selection. We compared his population to just a globally diverse population, and this peak at RMES1 popped straight out at us. We were able to, using his material, develop markers, and now those markers are being used in particular in U.S. breeding, also in Haiti, and in South America too, where this aphid is a huge problem as well too. And so if you want to read the specifics of this specific discovery, that's in 2022 Science Advances. So this happened, and we were excited and about the result, about the success of it, and we began asking why was this successful. And so I began kind of discussing with Gael all the ways in which he was breeding, to start to characterize his breeding program and see what we could learn from it. And so this is the diagram of his breeding program. So back when he started it, he started with just 24 elite founders from different breeding programs around the world, mostly African elite lines. He just started intercrossing with them, randomly intercrossing with them. And then he also incorporated a male sterility loci, so that it allows female and male assignment in his population, which then allows random mating between the two. And so what he does is he makes his selections about 8 to 10% and 4 to 5% of the different male and females, and then randomly crosses after those selections, randomly crosses the males to the females, and then continue to do this recurrent selection cycle, closing the loop at the F2 stage every time. When after had this population set up, farmers, realizing his success, wanted had different market demands. So there was a market demand for photoperiodism, having photoperiodic lines, and so in order to bring them into his population, they need to fit the idiotype of his population. And so what he'd do is he crosses the lines from his and by the unadapted lines that he wants to add in, makes F2 selections on that, and then those get added back in the population. And so this is basically showing that that allele migration for the conceptual slide that I showed earlier. So this is a breeding population. And so we began to characterize it genetically as well too. And so what this PCA plot is showing, the gray is just globally diverse panel, and his population genotyped at the same time with GBS markers is in blue here. And we can, this is what we expect. We expect to see, his population is locally adapting. It's becoming, allele frequencies are becoming going closer to 1. So they're clustering together. And we can see that for some specific alleles. RMES1, this is the loci that we discovered, and these are the markers that we developed, and we can see that in this population, those markers are at fixation. He's also, homogeneous for flowering time and height as a breeding program, with a market should be, and so for height. For the dwarfing genes, markers for them too, completely fixed for those. Flowering time genes in sorghum, the Mao genes are fixed as well. And then stay-green 5 is an important trait for staying green longer in the growing season. And he's also been selecting on that without markers up to this point. He's just been selecting on yield and fixing this QTL as well. So we're able to help describe his program, describe what's happening, what's required for local adaptation in Haiti, right? So now he has, based on that breeding scheme, he has this large breeding population in blue here. That's his main population, the recurrent selection population. This is, we were able to get sparse about 3,000 markers genotyped for about 5,000 lines in that F2 population. And so F2 and his main population here is crossing block. And so this is just showing he's got this large population, and then he's making F2 crosses and adding in this diversity. And so what we know is that what I just showed is that this blue population is fixed for this RMES1 trait. And these F2s now are actually segregating for it. And this is important because he needs, if nature wasn't doing the selection for him, the markers could do selection for him. And there are times in which the pressure is a lot lower during the rainy seasons. And so he can use the markers to make sure that individuals that are coming into the population have the alleles that they need to survive in the population, so that they're the new alleles at the other loci that they're bringing in don't just get immediately deselected out. And they're also again fixed for height and flowering time, which is important to be in this population. You need to have the homogeneous height and the homogeneous flowering time, and they're well headed towards fixation for the stay green as well too. So So we looked at this and we said, okay, well, we can do F2 mapping. We know that we expect that this RMES1, this RMES QTL is segregating. We expect to find it. We were right before, literally before we ran the GWAS, we also there was published a second QTL for this trait, and so that was also another prediction. We said, okay, well, is this QTL also in its population, and is it significant? And so, sure enough, we ran the GWAS and the F2s for two different survival at two different time points in the season, just a binary trait of whether the plant was alive or not. They went out and they measured that in the breeding program, and then we ran fit a binomial family term, and we're able to map. And sure enough, at early season, we can see that the second QTL that was just reported in the literature is significant. And then at the later season, both QTL are significant, and this is leading us to Carl Van Gessel, who will soon be defending his PhD in a couple of weeks here. Has been doing really nice work, kind of describing using Gael's program to describe the genetics of resistance. And so, for example, we can show that this at the early season RMES1 isn't what's maybe providing survival, but maybe RMES2, and then later both of them are conferring. And we're able to confirm this, that not only are they significant in the F2 population where they're segregating, but in his main population where the allele frequencies are much closer to being fixed for these traits, that it is indeed under selection. And then we can, for this and other traits, we can use the mapping combined with the scans. I just put FST scans, but there's other type of selection scans we can do as well too, just to prioritize new candidates as not only significantly associated in a breeding program, but also show that the breeder who's just been selecting for yield and surviving in the field under these different abiotic and biotic stresses, we can show that these alleles are working and favorable in a breeding program, which is important for breeding, is to be able to show that they actually work in a breeding program. So based on this relationship, we've termed the framework as flywheel genomics. So in engineering, a flywheel is used to regulate energy transfer from some sort of source to a motor. And in this analogy, in this framework, we're referring to, so genetics would be the flywheel for breeding, and breeding would be the flywheel for genetics. And so obviously there's no engineering thing that has two flywheels in a circle, but we think we like the analogy still. And so, right, the breeder is breeding, and we can use theory to understand where our allele frequencies should be. They provide the data, the observations. We work together to get those. Use genetics, we can use our tools. I'm a quantitative geneticist, we're using quantitative tools. And then we provide back information and tools. In this case, marker technology. We develop marker technology, and we provide information on and not only to the breeding program, but we get to publish on this and provide knowledge to other breeding programs as well too. This is an intensive collaboration. We are doing many things with Gael. And so one of the things we can do besides just developing markers is now we can actually track the alleles over the course of selection. And because he breeds in Haiti, he can oftentimes get 3 generations in a year. And so we can very quickly understand the dynamics of these different alleles. And one case we're doing that is there's very low vitamin A deficiency, like many parts of the world, is also an issue in Haiti. And so we're working to integress carotenoid alleles into the population, and so we'll be able to study how those alleles behave in a breeding program as well too. And then the more phenotypic tools that we can develop, they're starting to use to get a grant to use drones, and so if they can phenotype more, we can map more and we can understand more traits in this breeding program. Also, I think he breeds like 4 or 5 other crops, several of which he's been able to set up recurrent selection populations, and so most likely we'll continue to study in those crops as well too. And importantly for science, we get to study the dynamic nature of population development, which is breeding. And didn't forget about Africa. What we're learning here from Gael, we're now taking and working with breeding programs in Africa to set up these population development breeding schemes, so that we can then start to study and learn and traits that are important for Africa and African breeding in African breeding programs. Okay, so now present on some simulation. And so we want to do some simulation because we asked ourselves, like, why was this successful? And so the population design decisions that Gael made worked for both breeding and genetics. He's been pushing ahead his yield, and we've been able to study important traits in his population. So which decisions, was he making that made it this easier to do? And so just narrowing in on, I think one of the key ones that he did is high amount of intercrossing that he's able to do because of his male sterility system, and then the random mating. Even after selection, he's able to do a ton of intercrossing, and which is increasing recombination. And this is just showing that in the main population in blue here, but also even in these F2 populations that were created, we have pretty rapid LD decay and very low background LD levels in all of these as well too. So so that's an observation that's really pointing to the fact that this level of intercrossing is making it easier to breed, to do this type of mapping and understanding. And so, right, can we do this? So which decisions make it easier? Right, it's important that these decisions benefit both genetics and breeding. If you're just breeding and your goal was to make the best yield in line, you would make decisions mainly for genetic gain, and you could achieve new lines fairly fast, but that wouldn't work for genetics. And if you were just worried about genetics, you would spend the next 5 years making a nested association mapping panel, and it would have no use to breeding. But these decisions should work for both genetics and breeding. And so to kind of test that, we came up, this is I'm calling it the random mating scheme, and this is the breeding scheme that Gael has been doing, and we wanted to compare that to something similar. So I came up with this scheme that was practical. It's still recurrent selection, but there's no male sterility. So instead, we're making after selections, we're making our crosses, producing double haploids, so 100% inbred lines, making selections, and then after that, making all pairwise crosses. And so because of the all pairwise crosses, the practical decision to have a more stringent selection strategy is needed. But it also means that this strategy here it really is about recombination versus genetic gain. And so with a more intense selection strategy and then many crosses and then a 10 selection strategy, we expect that genetic gain would be very good, but not as much recombination as in this random mating scheme, or as a matter of intercrossing going on. So set out to simulate and compare these two schemes, use AlphaSim R. We simulated two traits. The first trait, highly polygenic trait, 100 QTL per chromosome, and then we wanted to simulate a new stress being applied. So we had a second trait, more allelogenic, just 10 QTLs, 10 chromosomes in sorghum. So 1 QTL per chromosome. And so the selection indices would look like this. Early on, the program is just selecting on the first trait, the yield, and then not selecting on the second trait in the dashed line here. And then when the new stress is applied, the selection indices needs to change. And so this the selection indices now has to favor the second trait and lower its selection on the other trait. And this is what happened in Haiti with the sorghum aphid. This is what may happen as climate change continues to shift, more biotype and other biotype shifts happen. And so the way we're going to evaluate the simulation results is, are they serving breeding? We expect genetic gain. Are they serving breeding and genetics? We would looking for maintaining diversity and variance at loci not under selection, and the ability to adapt to new and change in this changing stress, right? And so here I'm showing the phenotypic mean for these two traits in these two breeding schemes. So the blue being the random mating scheme, and the green being the pairwise mating scheme, the dashed line being the resistance trait, and the solid line being yield. And so we see before the stress happens, both programs are able to make genetic gain. The one that's favoring a higher selection intensity is making higher selection gains. When the new stress is applied, though, what we see is that the scheme that doesn't have that has more crossing has the ability to make gains on the second trait, whereas the one that's focusing on genetic gain more doesn't have the ability within the population to make gains on the second change. And so these are also, I ran these simulations over several times, and so I'm not showing the distribution, but it's actually fairly narrow distribution across the 1,000 runs of the simulation. Okay, so both are able to make genetic gains up until the stress, and then the one is not able to make genetic gains on the other one, and we might have some guesses about why that happened. And so there, because of this high selection intensity, linkage drag is reducing diversity and fixing non-selected QTL before they're needed. And we can see that from the simulation, even the trait that's not under selection is reducing in genetic variance. The other one is a little bit, but not even close to as much. And then once the selection for that trait starts, obviously the variance goes down. There's only 10 QTL. They get fixed pretty quick when they're selected on. And so this is main concept. Maintaining diversity in the population is it's easier than reintroducing it. And so this population, the random mating scheme doesn't have to introduce new diversity in when the new selection comes. We can just show that across, in simulation across the population, the minor allele frequency, that's what MAF is just continue to drop. And so just across the genome, due to selection on this one trait across the genome, you're getting a reduction of variance of diversity. Whereas when you have more recombination, you don't see that. And so like we did in real life, we ran a GWAS in simulation at generation 10, right when the stress was applied. And we can see that true positive hits for the random scheme are a lot higher. And then when in the pairwise mating scheme, and you might guess why that is, because the QTLs are being fixed. If there's no allelic diversity at a QTL. We can't detect it in GWAS. So I'll just point out that we're also adding to the simulation, all the different decisions that a breeder might be able to change, like where, when they're closing the loop, maybe F2, F3, we can ask those type of questions, and we are to kind of develop this framework. Also, what should your founders, your population look like in Haiti? With 24 founders, the breeder had both the positive alleles for resistance to a trait he didn't even know he needed until many years later. And so like how do you determine that starting founders? And so these are questions that we're finishing up asking, and then we'll be publishing on. And so I'll just kind of finish with some conclusions or my main arguments for this framework. And so the information from a breeding program can and should be used to do trait, genetic, and architecture dissection. And this, as I've argued already, is what is going to allow the information to be more readily accessible to plant breeding, is if it's done in conjunction with breeding. And I'm not unaware that sorghum was a tractable species for this. There's a male sterility loci that allows crossing. The trait that I mainly showed you today has a very easy phenotype to phenotype. It's either dead or alive, or wilting or not wilting, and is a diploid species, so the genetics are a little bit are simpler. So I'm not unaware of the tractability of working in this system, but my hope is that, well, the argument that I'm married to, the hypothesis that I'm married to, is efforts towards setting up this type of relationship will yield results. And so the efforts may be different levels of efforts depending on the species and the level of breeding program, where the breeding program is at. But if you want to have an impact in breeding for whatever species you work with, putting in the work to set up this relationship will yield results. And that's what I'm arguing. So and then we can think about these different principles of plant breeding and genetics, and which allow them to to work together. So and finally, this transfer of information doesn't work without coordination, doesn't work without collaboration. And so with that, I'll say thank you to the lab, my current lab, the crop adaptation lab at CSU, guided by Dr. Jeff Morris, and then a lot of the mapping work was done for Carl's PhD work that I mentored him through, and then this is actually an old picture. Some people have moved on, but there's many people in the lab that have touched this work or that continue to work with Gael's breeding program. A lot of what we do is moving towards working with breeding programs like Gael's, and then of course Gael and his staff, Yvens and Charles, and everyone that helped do all the sampling, and that has been great to work with. And then funding this type of research isn't these smaller farmers aren't being as intensively supported by commercial companies, by industry. There's not a return on investment for them, and so if we're going to have an impact in these farmers' lives, it's going to take public funding. And so these are the main funding sources for this project, but there'll be the Gates Foundation isn't on here, but they're a big supporter of this type of research, and can support our lab as well too. So, and with that, I'll leave my contact up here. Like I said, I'll be starting as a faculty at the University of Nebraska in January. Definitely looking for collaborations and students. I'm really looking for people that have a heart for this type for serving these communities, but doing so in academia, in plant breeding. And so it won't be all just plant breeding, it'll be plenty of lots of amazing discoveries that come out of these plant breeding programs to do research on as well. So with that, I'm happy to take questions.
Thank you, Brian. So do we have any questions from the audience in the room? Okay, so if nobody has a question, we can go home. No, we need to have a question.
Yeah, I can
So,
ask. Yeah, you said that this program prioritizes high diversity and high recombination. But how does that then work in combination with prediction? Because recombination and breaks up haplotypes and it gets difficult to estimate.
Yeah, great question. And right before I joined this lab, they also published on the recurrent the genomic selection that was going on in the program, setting it up. And so we know, so a lot of people want to do genomic selection in the breeding programs, but you actually need genomic selection really works best in this population development scheme. And so yeah, so with this level of recombination and diversity, they ran a simulation study kind of to determine if genomic selection would even beat out phenotypic selection, and it does. And it's just a matter of how often you need to retrain. And so from the when he closes the loop, Gael will pull out individuals that have that are diverse, but also representative of the population, and design his training population around that. And so he does from this large population create a training population, which then gets retrained every so many generations, and it's working. We see high prediction accuracies, and we see and the resulting genetic gain.
Do you have any idea of what the effective population size of such population is?
Yeah, I don't have an exact number for you. It's, you know, it's probably not huge with the amount of founders that he started with. So yeah.
Thanks.
Thank you. So you were talking about how this is dynamic population, so we should have kind of dynamic process of doing this thing that you showed, like mapping. So how often do you actually need to do this analysis, this mapping, this selection of these SNPs?
I mean,
markers.
Yeah. I mean, if the right training is provided and the right pipelines are developed, I mean, every time you collect genotypic and phenotypic data, you can do the mapping. For a trait like this, for example, you know, we probably don't need to keep redoing the mapping. If there are new QTL that come out, we can identify them in this program. Rather, we're more interested in just tracking the alleles, and then we can, if we're worried that you know, there might be negative pleiotropy, for example, maybe a yield effect, then we can definitely remap and maybe do some evaluation on that. Yeah.
Okay.
Yeah. So
you,
I think it's trait dependent on how often you actually you want to map, and it's a great question, one that I definitely will be part of my lab and thinking about is how do we actually study dynamic processes in dynamic populations, and what even is a dynamic process in genetics, you know. So
yeah, thank you very much, Brian. I see we have no questions online, so I would like to thank you one more time to joining. Hope you will be part of CDDG in future as well, and wish you nice rest of the day.
thank you everybody.